Howdy, our names are Cassandra Fernandez and Alondra Curiel, and today we're going to present our lab report 13, PCR and gel electrophoresis. Introduction. In this lab, we use PCR, restriction digestion, and gel electrophoresis to detect specific DNA sequences in food samples. PCR amplified the target region from each sample, and the restriction enzyme then cut the DNA only if the pathogenic sequence was present. Gel electrophoresis separated the resulting fragment so we could see the banding patterns and compare them to a DNA ladder. This allowed us to determine which samples contain the pathogen and evaluate food safety. Materials. For the reagents, we had master mix, primer mix, CBE buffer, restriction enzyme, which was diluted, Aragos gel, fast DNA ladder, and then for the equipment, we had the mini PCR thermal cycler, the incubator or heat block, electrophoresis chamber, the micropipette, sterile pipette tips, UV blue light transluminator, and the microcentrifuge. For the samples, we had non-pathogenic control DNA, pathogenic control DNA, lettuce DNA sample, tomato DNA sample, PCR products from tubes 1 through 4, and digested samples from R1 through R4. For the procedure, part 1, which was the restriction enzyme, we labeled 4 new 0.2 milliliters tubes as R1 through 4. Then we transferred 15 microliters of each product, PCR product. So for tube 1, it was R1, and then for R2, tube 2, it was R2, and then so forth. And then we added 2 microliters of diluted restriction enzyme to each one, closed the caps, and flipped to mix. Then we split in flicks to get the liquid down, and then we incubated it at 37 degrees Celsius overnight. For the part 2, which was the gel electrophoresis, we placed prepared gel into the chamber. We added a buffer just enough to cover the gel. We loaded wells using a new tip each time, and then lane 1 was 10 microliters of the DNA ladder. Lane 2 was another 10, which was the R1, and then lane 3 was R2, lane 4 R3, and lane 5 R4. And then we ran the gel for 20 minutes, and then we visualized the DNA using blue light or a transluminator, and then we kept running if bands aren't well separated. Then we took a photo and compared the sample bands to the ladder to estimate fragment sizes.
Now for the results. This is what our gel looked like. And for R1, which would be this one right here, we can see a bright band. So that would be a non-pathogenic. For R2, right next to it, we see about 2 to 3 bands. So then that would be pathogenic. For R3, we see one bright band, and that would be non-pathogenic. And then for our R4, we see 2 bands, so that would be pathogenic. In conclusion, the results from this lab clearly showed how PCR, restriction digestion, and gel electrophoresis work together to detect pathogenic E. coli in food samples. The restriction enzyme only cut DNA when the pathogenic sequence was present, which resulted in 2 separate bands on the gel. Samples that remained uncut appeared as a single band, indicating non-pathogenic DNA. Based on the banding patterns, the lettuce sample R3 showed 1 uncut band and was non-pathogenic, while the tomato sample R4 showed 2 distinct bands, confirming the presence of the pathogenic gene sequence. These findings demonstrated how molecular tools can accurately identify contamination sources and help evaluate food safety in real-world scenarios. Thank you for listening.